This is an overview of an article entitled Smoking is Associated to DNA Methylation in Atherosclerotic Carotid Lesions, published in the September 2018 issue of Circulation, Genomic and Precision Medicine. It has been shown that aberrant epigenetic modifications are involved in the pathophysiologic pathways of numerous diseases like cancer and neurodevelopmental disorders. Mechanisms such as DNA methylation or DNAm that provide regulatory information to the genome, therefore influencing gene expression without altering the primary nucleic acid sequence, are referred to as epigenetic modifications. DNAm is a heritable epigenetic mechanism whereby methyl groups are added to the cytosine or C or adenine or A nucleotides in the DNA molecule. In humans, the most common DNA methylation is at cytosine and CpG dinucleotides. Epigenetic alterations can be influenced by age, environment, genetic variation, and lifestyle choices such as tobacco smoking. Tobacco smoking is a major risk factor for the development of atherosclerosis and subsequent cardiovascular disease. Smoking induces endothelial dysfunction and vascular inflammation. However, the exact pathophysiologic mechanisms are not fully understood. Epigenome-wide association studies, or EWASs, have identified changes in the DNAm patterns within blood cells that are associated with smoking. Scrutinizing the effect of tobacco smoking on DNAm in the actual vascular lesions themselves might therefore provide important insights into vascular pathology. In the study under discussion, a two-stage epigenome-wide association study of atherosclerotic plaque-derived DNA methylation with current tobacco smoking was performed in carotid endarterectomy, or CEA, patients from the AtheroExpress Biobank study. First, a discovery study was conducted where DNAm at commonly methylated sites, namely CpGs, was assessed in atherosclerotic plaque samples from 485 CEA patients. Replication of the smoking-associated methylated CpGs in atherosclerotic plaques from the discovery study was attempted in a second sample of 190 CEA patients to validate the findings of the first study. To gain better insight into the tissue specificity of the methylation results obtained in atherosclerotic plaques, the authors also performed an EWAS in blood samples from the same patients, and they carefully scrutinized literature investigating such changes in blood or other tissues. These Manhattan plots illustrate the association of DNAm in carotid atherosclerotic plaques with current tobacco smoking during each stage of the study, with the top left panel representing the discovery study, the top right panel the replication study, and the lower left panel the results of a fixed effects meta-analysis of the discovery study and replication study. In each panel, the epigenome-wide significant CpGs are depicted as gray dots. In the meta-analysis, the authors found that tobacco smoking was significantly associated with differential DNAm in atherosclerotic lesions of 4 CpGs mapped to two different gene loci, AHRR and ITPK1. All of these 4 CpGs showed reduced DNA methylation in current smokers as compared to former or never smokers, as represented in the box plots shown here. The strongest associations were found for the 3 CpGs that mapped to the AHRR gene locus, AHRR encoding a repressor of the aryl hydrocarbon receptor transcription factor involved in xenobiotic detoxification. Differential DNA methylation at this locus has previously been associated with smoking on numerous occasions and in various tissues. The other smoking-associated CpG locus is located near ITPK1, a gene encoding inositol tetrakisphosphate 1 kinase. Differential methylation at ITPK1 has also previously been implicated in smoking. Here's a Manhattan plot depicting the association of DNAm in blood cells with tobacco smoking, with the epigenome-wide significant CpGs shown. The authors identified 17 significant CpGs in 89 blood samples derived from the same patients, mapping to 8 genes, all of which showed lower DNA methylation in current smokers compared to former or never smokers. Of these 17 CpGs, 8 have previously been associated with smoking in blood and other tissues. Of note, the 4 smoking-associated CpGs identified in carotid atherosclerotic plaques in this study were observed to strongly replicate in the blood cell EWAS. Even though strong associations were identified in the paper under discussion, there were some notable limitations. The analyses were based on patients' current smoking behavior, which would not have reflected time-dependent effects of smoking on plaque methylation. DNAm was measured in a rather heterogeneous tissue and not in specific cell types, possibly explaining the limited replication, namely only 4 out of 10 CpGs being significant, and why no associations between the smoking-associated DNAm and specific plaque characteristics were found. In addition, the study comprised patients with advanced atherosclerotic disease, which might also explain the lack of associations with plaque characteristics. Finally, the replication cohort was of limited size, reducing power in the meta-analysis. In conclusion, tobacco smoking was associated with DNA methylation at 4 genomic sites in the carotid atherosclerotic lesions of CEA patients. The combined results of the experimental data from this study and a literature search suggest vasculature-specific methylation differences induced by tobacco smoking. Future studies should verify these findings, and they should focus on the importance of investigating DNA methylation in the vascular lesion itself, rather than just proxy tissues like blood, as well as the underlying mechanisms of AHRR and ITPK1 methylation in the vasculature as a response to smoking.